Welcome to AP Biology. Hi, I'm Gail Fizer from Darnell Cookman Middle High School and Florida State College in Jacksonville, Florida. A lesson overview for today. Today is the last topics for Unit 6, Gene Expression and Regulation. I know we're all doing something different today than we would have thought months ago. Today, right this minute, I should be at the University of Florida watching my son graduate with his master's degree and planning a party, but that happened anyway, but we're going to do it online. And I'm sure that you're not doing what you thought you would be doing today. But if you're listening and working and we're all safe, that's what's important right now. All right, topic 6.7, mutations. We've covered all of this already. So I'm just going to tell you, whatever something comes up, I'm going to tell you where to reference that so we can spend our time on biotechnology. By the end of this lesson, you will describe various types of mutations, explain how changes in genotype may result in changes in phenotype, explain how alterations in DNA sequences contribute to variation that can be subject to natural selection, and explain the use of genetic engineering techniques in analyzing or manipulating DNA. So you'll need pen and paper for a few questions today. So our warm-up is from our homework yesterday with Ms. Kate, and it's from gene regulation. And this is a very rich question because it has a lot of reviewing other things in the question. So human cells divide every day for normal growth, replacement of cells, and tissue repair. So right there we have the function of mitosis, right? The dividing cells proceed through 3 phases of the cell cycle. We have interphase, mitosis, and cytokinesis. When a person experiences a cut, damaged cells secrete a protein called epidermal growth factor. So this can be our skin, and that is our ugly cut. Okay, I'm going to draw some epidermal cells because I'm going to need them in a moment. Those are very big epidermal cells. Okay, so when a person experiences a cut, damaged cells secrete a protein called epidermal growth factor. And let me draw that in, okay? All right, epidermal skin cells in the vicinity of the injury possess proteins in their cell membranes called epidermal growth factor receptors. So here we have receptors on these cells, okay? So now this protein, and there's the protein, and there's the protein receptor. Okay, cancer can result from disruptions in the cell cycle control. Mutations that increase the production of EGFR have been associated with skin cancer. So we're going to be, if we're talking about cancer, then we're going to be, cells are going to be dividing out of control. And this cancer was at the head and the neck region. So what are we going to do? We're going to describe how a mutation outside of the coding region. Now remember, just yesterday, this is a gene, kind of crooked gene, and here is outside. That was our coding region, and this is outside of our coding region. Okay, outside of a coding region of a gene that encodes EGFR could lead to the overproduction of EGFR protein in a skin cell. So if you're just joining us for the first time, I'll give you a few seconds. Let me turn my timer on anyway. And if you did your homework yesterday, then you should have this ready. All right, now we can start. All right, so here are some options for answers. Mutation in regulatory sequence or promoter or enhancer could increase rate of transcription of EGFR gene. Okay, that's the answer that I would have written down. Here's some other options though. Mutation in transcription factor could increase production of messenger RNA, which would lead to production of more polypeptide. Gene duplication may increase production of EGFR. Gene translocation may inactivate the repressor. Mutation in repressor gene may remove controls on cell division, and change in messenger RNA sequence may increase translation efficiency or messenger RNA stability. When I wrote this to prepare for today, this is the option that I wrote before I read the rubric. Okay, so what you need to know. Okay, so there are a lot of these slides that are just review information, and I'm just going to kind of skate quickly through those. And if I need to stop for a moment, I will. But if it's old information that we've heard before, then I'm going to refer you to where you should go to re-listen to that if you've been following on the videos. All right, the function and amount of gene products determine the phenotype of organisms. The normal function of the genes and gene products collectively comprises the normal function of organisms. Disruptions in genes and gene products cause new phenotypes. This is a great, there's a question that follows this. So this is a great example. Example of a change in a genotype that results in a change of a phenotype, okay? Mutations in the CFTR gene disrupt ion transport and result in cystic fibrosis. A normal channel allows the transport of chloride out of the cell. The increase in the intracellular chloride concentration causes water movement into the cell due to osmosis. So here we're bringing in osmosis into this along with protein transport. The mucus is dehydrated and sticky and builds up in multiple organs. So let's look at what should be happening. So here's our chloride channel, okay? So here's our chloride. It should be able to move through to the extracellular fluid. But yet, with this mutation in this protein, so here's our channel and it's mutated. Now this chloride ion cannot get through this channel. So it's not moving through, but what's happening is that the solute inside the cell is increasing, which is making the water potential lower. So the water is going to move from the outside to the inside through aquaporins. Few will get through the phospholipid bilayer, but through aquaporins, the water will move through. And then this mucus is going to, which had a great deal of water and was hydrated, is now going to be dehydrated and sticky and start affecting multiple organs, okay? All right, so here's a question about CFTR protein. It's made up of 1,480 amino acids linked together in a chain. Some humans produce a version of the CFTR protein in which phenylalanine, an amino acid, has been deleted from position 508 of the amino acid chain. Which of the following best predicts how the amino acid deletion will affect the structure of the CFTR protein? So let's take a look at these options. A, it will have no observable effect. Well, on the structure of the CFTR protein, well, I just showed you what was going to happen with this, so we already know that. It will affect the primary structure of the CFTR protein. That's true because this was amino acids, right? So we had a strand of amino acids and then in our primary structure, and now one's gone, all right? Or it's substituted out for the wrong one. But the other levels of protein structure will not be affected. That's not true. The properties of that amino acid then change the structure and the function of that CFTR protein. So that's not it. It will affect the secondary, tertiary structures, but the primary structure will not be affected. That's not true. We just said that at that position that there had been a deletion. Yes, a deletion. It will affect so that's not it. It will affect primary, secondary, and tertiary structures. Yes, it affects all of those levels of protein structure. So D is our answer. Let me check. Yeah. All right. So now we have another mutation. Alterations in DNA, I know you've seen this before, in a DNA sequence can lead to changes in the type or amount of the protein produced and consequent phenotype. DNA mutations can be positive, negative, or neutral based on the effect or lack of effect that they have on the resulting nucleic acid or protein and the phenotypes that are conferred by the protein. So this again is in antibiotic resistance. I think I had the same diagram in natural selection the second night that we started the videos in natural selection. But anyway, you have bacteria in your body. Some of them are going to be resistant to antibiotics. And so you take the antibiotics, they kill all those that are not resistant, and then the resistant reproduce, make more resistant, and then they also can even exchange in a horizontal exchange to other bacteria. Okay, I guess that would be positive I guess the perspective, that's probably a positive move for the bacteria, but a negative move for the human being. All right, so sickle cell was another mutation that we've talked a lot about. In normal, remember that we had a T, and we that it coded for glutamic acid, and then the mutated hemoglobin, we have an A, and it coded for valine, and that was a problem because the glutamic acid was hydrophilic and valine is hydrophobic, and it ended up getting these hemoglobin molecules that stick together in this fiber form, and then they end up in this structure. Now that would be a negative effect, is if we had two recessive alleles that do this. We'll talk about that. So if we have, we're just going to talk about this top part. If we had two recessive alleles, then that's negatively going to impact this individual because they're going to have sickle cell disease. If you have no sickle cell, if you're not a carrier, then you are subject to getting malaria. And then if you are a carrier, then you have the heterozygote advantage. You're not going to get malaria and you're not going to have sickle cell. So there's a positive experience or positive mutation in this case. All right, changes in genotype may result in changes in phenotype. Errors in DNA replication or DNA repair mechanisms and external factors, including radiation and reactive chemicals, can cause random mutations in the DNA. Ms. Kate just talked about this 2 days ago in DNA structure and function and replication. Whether a mutation is detrimental, beneficial, or neutral depends on the environmental context. Mutations are the primary source of genetic variation. If you need to review DNA replication or repair mechanisms, it's in topic 6.1 and 6.2. All right, errors in mitosis and meiosis can result in changes in phenotype. Changes in chromosome number often result in new phenotypes, including sterility caused by triploidy and increased vigor of other polyploids. So triploidy, we're talking what we could be talking about Down syndrome, and polyploidy, that could be like strawberries. Changes in chromosome number often result in human disorders with developmental limitations, including Down syndrome, trisomy 21 that we just mentioned, and Turner syndrome. So if you need to revisit changes in chromosomal changes or environmental effects on phenotype, then go back to 5.5 and 5.6 or where those topics are located. All right, changes in genotype due to alterations in DNA sequences may affect phenotypes that are subject to natural selection. Genetic changes that enhance survival and reproduction can be selected for by environmental conditions. So this is in Unit 6, okay? This is talking about natural selection, and evolution, and especially natural selection, has been addressed in different parts, minor parts, listed in units 1 through 6. So I highly recommend that, for the sake of natural selection, that you go back and look at our first 2 videos. 7.1 and 7.2 are both about natural selection, and they could be helpful in understanding some of this biology. All right, so now we get to some topics that we haven't discussed in any previous video. So transformation is a method of horizontal acquisition. So what that means, so if I was a bacteria and then I have through binary fission, I made an offspring. Okay, that's not what I'm talking about. I'm not talking about DNA going from this to my offspring. Okay, that's not it. So what I'm talking about is a bacteria. In this case, we're talking about transformation. We're going to have a bacterial cell, okay, which is going, some genetic information is going to end up, that was here. Okay, let's make an example of this right now. Okay, so some genetic information that was in this bacteria, and it's going to be given to its neighbor that doesn't have it, okay? So it was like that, and then it got the plasmid. And we'll talk about a number of ways to make that happen. All right, so let's see, what else? let's talk about transfer. Oh, this is transformation. This is from students, Petri dish in my classroom this year. Okay, so I'm gonna talk through the process, and I drew this at, and I'm on the Eastern Standard Time, and so I drew this like 4 o'clock this morning. So it's not beautiful, but I think it gets the point. All right, so we have some E. coli, we have E. coli bacteria growing, we have some colonies, and we're going to take a few small colonies and we're going to mix it with calcium chloride, okay? And we're gonna keep this cold while we're working with this. And then we're going to add a plasmid, and this plasmid is going to have genes on it. And so what this plasmid has, it has a gene that will break down arabinose. It has a gene that will turn it green with green fluorescent protein. It also has a gene that will give it ampicillin resistance, okay? So now we're going to put this plasmid into the tube with our bacteria sample and our calcium chloride, and we're going to keep it on ice very cold, okay? Now, we're going to take it from the cold to the heat, and we're going to do what's called a heat shock. So we're going to heat shock it, and after we stay in the water bath for about 50 seconds, we're going to go straight back to the ice, okay? Now, what happened during that time period? Let's say this is our phospholipid bilayer when we're very, very cold, okay? We are very close together, and it's cold. All right, and now we're going to get put into some seriously warmer than we already are water. Okay, so let's get rid of this. And then what do you think is going to happen to this phospholipid bilayer? Okay, so now we're going into, excuse me, into warmer water. And now there's going to be not so tight, okay? There's going to be some room. It's more fluid. Now, this is the opportunity for the plasmid to get through this phospholipid bilayer. But the plasmid has what? It has a negative charge. So the calcium chloride, which has a positive charge, is actually going to be like the Secret Service and get kind of camouflage, make this not so negative and be able to shuttle right through this membrane, okay? So that's the hopes, that we're going to get it, we're going to expand the plasma membrane, and then we're going to escort this plasmid through there, and then the plasmid will be inside the cell. Okay, so now that the plasmid's inside the cell, it can be expressed to make other proteins. All right, so how does this work? We have to have operons that were talked about yesterday in gene regulation. So this is their arabinose, okay? And arabinose was in one of the plates. And arabinose is what's going to bind to this enzyme that's going to bind to this promoter area that's going to enable RNA polymerase to transcribe the messenger RNA that's going to go to the ribosome to build the enzyme that's going to break down the arabinose for a nutrient, okay? Now, at the same time that all that's happening, we also have this GFP. So in the promoter area where this arabinose was binding to allow this RNA polymerase to transcribe, it's also going to transcribe for GFP. So we're going to break down arabinose, and we're going to be able to turn green in the process. Now, it also has a gene on it for ampicillin resistance. So the ampicillin resistant gene is turned on, and it makes a protein that's going to then break down ampicillin so the bacteria can grow. All right, so now that we have all of that behind us, we're going to put on the plates, we're going to put two plates. This has plasmid, so there was some transformation that hopefully had occurred. So we're going to put this sample on the plate with the plasmid on an LB, which is Luria broth, and agar, and then ampicillin, which is an antibiotic. And the same thing here, LB, ampicillin, and then the arabinose sugar is in the Petri dish. Now, this has a plasmid also. We have here no plasmid on this plate, and no plasmid on this plate. So the results in the next 24 hours, we have some isolates growing right here. And so that means that we have some transformation, right? But they're not growing green because the arabinose was not there that turned on these two, okay? All right, so now we have, in this plate, we have arabinose, and so now we have, this was able to be turned on. We transcribed GFP and the enzyme to break down the arabinose, and now we have the green glowing fluorescent protein, okay? Now, we didn't have a plasmid in this, but yet we put it in ampicillin, which is an antibiotic, and so it killed all the bacteria. Now we put one without the plasmid in this plate, but it's only on LB, it's not in the ampicillin, so it grew all over the plate, and that's called a lawn. Okay, so that is transformation. We took non-green, okay, glowing, they were not glowing green, and now they are. So we transformed them, we gave them genetic information that was able to be transcribed for a new phenotype. Okay. All right, so this is from, actually these are plates because they were just beautiful from I teach a college class, adjunct professor for our Florida State College, and I teach introduction to biotechnology and bioinformatics there. And these were plates, I do this lab in both of those classes, my FSCJ classes and my AP classes at Darnell Cookman. And on this particular, mine looks crazy right here because I get the wonderful opportunity to do this a lot. And so I always change it up. And I think I put extra plasmid in my solution on that one. That's why it was so many. So in my AP classroom, I always follow up a lab with this particular lab because it's so, the novelty of it, and it's so many steps, and you need to not only know how to do the lab and make the procedures go right, but to have understanding of what you were doing and why you were doing it at the end. That this is a set of questions that I always give them. So I'm gonna give them to you about what we just talked about. In a transformation experiment, a sample of E. coli bacteria was mixed with a plasmid containing the gene for resistance to the antibiotic ampicillin, and it says AMP and a little r. Plasmid was not added to a second sample. Samples were plated on the nutrient agar plates, some of which were supplemented with the antibiotic ampicillin. The results of E. coli growth are summarized below. The shaded area represents extensive growth of bacteria, and dots represent individual colonies. So this is a lot of growth, and this is just a few colonies, and this is nothing, okay? All right, in a second experiment, the plasmid contained the gene for human insulin as well as the AMP-R gene. Which of the following plates would have the highest percentage of bacteria that are expected to produce insulin? Okay, now, it's a trick question kind of, okay? Because now the highest percentage of the bacteria that are growing, well, 100% of these bacteria on this plate have been transformed, okay? Now, because they have in ampicillin and they have the plasmid. So they all have the gene for ampicillin resistance. So that they have the gene for ampicillin resistance, then they also have the insulin, okay? So this would be 4 only, which would be C, okay? Now, the next one, plates 1 and 3, were included in experimental design in order to, okay, here's 1 and 3, both of them have growth all over them, and they don't have ampicillin. So let's look through our choices. They were in order to demonstrate that E. coli cultures were viable, demonstrate that plasmid can lose its AMP gene, demonstrate that plasmid is needed for E. coli growth, or prepare the E. coli for transformation. So I hope you chose A, right? That to make sure that E. coli could grow without the treatment. All right, plates that have only ampicillin resistant bacteria growing include which of the following? So only ampicillin resistant. So we have to have ampicillin, there's nothing growing here. So our only option is 4, okay, which is C. Which of the following best explains why there is no growth on plate 2? The initial E. coli culture was not ampicillin resistant. The transformation procedure killed the bacteria. nutrient agar inhibits E. coli growth, or the bacteria on the plate were transformed. There's no growth here. So your answer? Okay, it's A. Yes, the initial E. coli culture were not ampicillin resistant. And then which of the following statements best explains why there are fewer colonies on plate 4 than on plate 3? So why are there fewer here than there? Okay. All right, so plate 4 is the positive control. Not all E. coli cells are successfully transformed. The bacteria on plate 3 did not mutate. The plasmid inhibits E. coli growth. So I hope you said that not all E. coli cells have successfully transformed. And that's transformation efficiency. Remember, we did transformation efficiency with something else just maybe a few weeks ago. Anyway, the amount that did divided by the amount that could, and then you could have your percent that were transformed. Okay, so here's another way of horizontal acquisition. Okay, so we're going to take a virus this time. We're still going bacteria DNA to bacteria DNA. So here's our virus, and our virus has, let's give him pink. Okay, he has pink DNA, and he's inserting it into this bacterial cell. This is a bacteriophage. Anyway, inserting it into this bacterial cell, and he is using all the machinery. There's some ribosomes in the DNA. This whole cell is just getting destroyed and used. Anyway, let's make this purple. Here's some of the bacteria's DNA. And so now what is the purpose of this virus? His goal is to make more virus. Now, if we have in this mix, this happened, okay? And this virus is going to leave with bacterial DNA instead of viral DNA. And now this becomes incorporated into the genome of this bacteria. So we went from this bacteria to this bacteria, and that's a horizontal acquisition also, okay? And all of these increase genetic variation. Okay, here's a question, and we're adding in a little, here's transduction, which we just talked about, plus fertilization. All right, which of the following correctly compares the two processes shown above? Both processes increase genetic variation. Yes, they do. Both processes represent aspects of sexual reproduction. No, this one does, but this one does not. Both processes require partitioning of genetic material in organelles. No, the bacteria does not have a nucleus and doesn't have compartmentalized partitioned organelles. So no. And the amount of genetic material per cell remains constant in both processes. No, it does here. Well, there's an addition here, and there's a 1N to a 2N condition here. So the answer here is A. All right, here is conjugation, and this is bacterial cell to bacterial cell. And so if we have, I'm going to put our one singular chromosome in here, and then I'm going to also add, I'm also going to add a, that's not really good, you can't see it. Let's do this. Okay, so here's my plasmid. Now this cell does not have a plasmid, but if this cell, that this plasmid is replicated from this cell, it can be put into this other bacteria through what's called a conjugation pilus that is formed with some small proteins between each of these two bacteria. That's conjugation. All right, here's some horizontal acquisitions, and there's two ways you can do this. This is called transposition, and what happens is you can have a now let's do this one. This one is going to be cut and paste, and this one is copy and paste. So here we have this particular segment of DNA, and it's going to be copied, and then it's going to be inserted somewhere else in the genome. Okay, so now it's going to be there twice. And we're also, in this case, we can cut it out and totally, it's not there anymore, and then insert it to somewhere else in the genome. And that's called transposition. It's also a horizontal acquisition of genetic material, and it also increases variation. All right. Let's see, I cut out something, that's why I kind of paused. In the regular PowerPoint, there's a question that has to do with eukaryotic cells and transposition. I cut it out due to time, but it's there for you to work on when you have time. All right, related viruses can combine, recombine genetic information if they infect the same host. So look at this. Here we have an avian influenza virus, and here's RNA from that virus. And here are the proteins on the envelope of that virus. And then we have a human influenza virus, and we have RNA molecules, and there are the proteins on the coat of that virus. Now, this pig respiratory tract cell has receptors for both the avian influenza virus and for the human influenza virus. So both of these are able to enter the cell. Now this is the pig's DNA, which has nothing to do with what we're about to talk about. So now this new virus, this new strain of virus can be formed, and it has both, look, it has the avian and it has, what's my color? And it has the human, okay, the human influenza. And then we notice that it does have the avian flu proteins on the outside. Okay, so this is how both of these got mixed together in this one cell and created a new strain. Very simplified version. All right, so here's a question about that. The cells lining the respiratory tract of pigs have receptors for both avian and human influenza viruses. Based on the model above, which of the following best describes the origin of the new strain of human influenza virus? The new viral strain inherited a mixture of genetic material from both avian influenza virus and human. Yes, it did. The new viral strain inherited RNA molecules from the RNA and packaged them inside the human influenza virus membrane. No. The new viral strain inherited a mutant DNA molecule from the pig. No. And the new viral strain inherited an RNA molecule that had been recombined with the DNA of the pig. No, it was both the avian and the human. So what I like to do with, when I have, so if this is in the CED and I want to know what the people who wrote the CED, what the people who were writing the questions that support the CED, how all their minds are working together. Like, what do these words not only mean to me, but what do they mean to the person writing questions or to the team writing questions? And so that's why I do a lot of this multiple choice work, is to see where they're coming from with this information. So this was built from a statement, that statement in the CED. So I hope that's helpful. That's how I try to figure out what exactly they mean if I feel like it is vague or maybe needed some more details to tell me what I need to know, but that's how I figure it out. All right, reproduction processes that increase the genetic variation are evolutionary conserved and are shared by various organisms. So here we have a diagram: crossing over, conjugation, and fertilization. The question asks, the processes illustrated in the models depicted above all result in which of the following? So transcription, an increase in genetic variation, yes. An increase in chromosome number, where is that happening? That's happening just in this one, right? And horizontal gene transfer, that's happening just in this one. So that's not it too. So our answer, all of these that we've been talking about, increase in genetic variation. So let's talk, what did we talk about? We talked about transformation. We talked about transduction. We talked about conjugation, transposons. Okay, all of those work in the same way, horizontal genetic transfer. All right, so now let's talk about some biotechnology techniques, some genetic engineering techniques. Polymerase chain reaction, we'll talk about that first. It stands for PCR. And what it does is it amplifies a copy of DNA, which means it's going to take usually a gene of interest or a certain fragment of DNA and make 1 million copies out of it. This is my, I don't know where that did. This is my PCR in my room, and I have, it has this 25 little slots here, and what these are 0.2 ml PCR tubes. And what's going to happen is that we need to make more copies. So we're going to do a synthetic version of what happens in replication, okay? So what has to happen in replication? The two strands have to separate. So to do that, then we're going to have to denature it. And we're going to denature it at a high temperature of 94 to 96. So now we're going to get those strands to come apart of that double-stranded DNA. And then we're going to have to put a primer on. So we're going to have to cool it down for it to have the primer put on. It's called annealing. And then we're going to heat it back up just a little bit for the nucleotides to be put together on the new strand. So we go through a little cool down and a little warm up. And we have to do that through several cycles. This is a setting. It had 35 cycles, so I could end up with about 1 million copies of particular DNA fragments when we got through with running the PCR. Okay, now we're going to talk about gel electrophoresis, and now we can, after we have amplified this sample, now we can separate it as far as fragment length, and we could do charges also. And so this is in my classroom, and this is a gel electrophoresis apparatus or tray. And what you see here is a negative N, and this red is a positive N. And then we're going to attach it to some power. And what's going to happen is that there's going to be hydrolysis occur, and then there's going to be a movement from, but I'm going to show you how in just a moment. If you put negatively charged molecules on one end of the negative end, then they're going to be attracted to the positive end. So they're going to move, okay? This is just practice before you learn how to use DNA, which is expensive, with food coloring. Okay, so we have a lot of things to talk about on this to kind of, if you've never done this before, never talked about this before, I'll do my best to make this quick, but as thorough as possible too. So in order to separate fragments, we're going to need to cut DNA into fragments. And there are many restriction enzymes, and they're made from bacteria. And bacteria produces a restriction enzyme in order to destroy viral DNA that's invading its body. Okay, so if we were to cut with this restriction enzyme, this particular segment of DNA, it cuts at a palindrome. So if you can see, we have a GAA-TTC, we have a GAA-TTC. And so this came from E. coli, and it was restriction enzyme number 1, okay, that was discovered. And so this is the site that this restriction enzyme always cuts at. So so we're going to be dealing with these 4 individuals. So if we had 4 individuals DNA, okay, and we were to cut it with restriction enzyme, we may cut, this person may have that palindrome there, there, and there. This one may be here. This one may be only there. And this one could be in many different places, all these different individuals. So what happens is these are cut into these fragments that are these lengths. Now, what a fragment is a number of base pairs. So if you had 1, 2, 3, 4, 5, whatever, 8 I have there, that would be your base pair number, okay? But they're in the hundreds and the thousands. So we could start off with fragments that are 30,000 base pairs long. So now we're going to load them into what is a well at the end of these gels, okay? And what it is, was made with a little comb, and it's a depression, and you're going to put your DNA sample into it, and it's going to be, DNA is negative, so we're putting this on the negative end, and then this is the positive end. And when this is turned on, the fragments are going to move. Okay, they're going to move towards the positive end. But there are different lengths of fragments. There's very long ones and there's very short ones, and they have different numbers of base pairs in them. So what's going to happen is, and I describe this to my students, if we all went to the mall shopping, and each one of us, of the 20 something of us in class, we all held hands and we tried to walk through the crowd in the mall. We're not going to get there very quickly because there's little places for us to get through, but we're not going to fit through there at any fast rate of speed. Okay. But if there were just two of us, then we would be able to move through the pores through to get to the other end quickly. So that's what's happening here. The smaller fragments, here's the smaller fragments. They are getting through this porous agarose and able to get to the other end faster. So this could be like 30,000 base pairs long here, and then 20 and 10,000, and then 1,000 and 500, and then 100, just making up numbers. Okay. All right. So I hope that helps with what restriction enzymes and gel electrophoresis does. So this is some UV transilluminators. And since the college that I work at also is like 5 miles down the road, I have the ability to jump in the car on planning and run down our samples so I can take pictures. And so this is a real huge desktop transilluminator. And I think we were working on PTC. That's the bitter taste lab when we were doing that, looking to see if we were homozygous or heterozygous for a trait or no trait. And this had to do with a food poisoning lab. This is one of my favorite things to do. Anyway, it's the same thing on both of these slides, but I wanted to, instead of just showing you how these look when you separate these fragments, I wanted to address something that I have to give a shout out to Maya. She asked me to talk more about positive and negative controls. And this particular lab is where it all comes clear because there's so much other things that can go wrong. So a positive control receives treatment with a known result. Okay, we know what it's going to look like. And a positive control is a control for what we don't know can happen. So if we, let me see how I can do here. So I think, and I didn't go back and look because everything's at school, but I think that this open place right here where there was nothing, I think that was our negative control, which had no treatment at all. I'm pretty sure that was what that was. And then I believe this was our positive control. So our positive control is there to show us what a positive looks like. And then I believe this was the match for the positive control. So let's say that none of these, let's say there were no results, okay, that this was not there. Could we say that was negative? Yes, if we have this positive control to show us what a positive control, then we can say this is negative. But what if this positive control was missing? Could we say this is negative? No, we couldn't, because probably if this is missing and it should be showing a positive result, then there's been some type of operator error. And this is one of the labs where that's so easy for that to happen. And when you're working with DNA and pipetting and gel electrophoresis and all that. So if everything was blank and even our positive control was blank, then we knew one of these other things out of our true control that was happening. I hope that makes sense. All right, so let's do some work with this. All right, so we have a gel electrophoresis tray. Which of the following statements best explain the pattern seen on the gel with regard to size and charge of the molecules of A and B? So this is I've done this before and put the wells in the middle and started with our DNA sample here, and then waited to see maybe with food coloring or some other type of something in my room, but not with DNA. Anyway, which of the following statements best explains the pattern seen on the gel with regard to the size and charge of the molecules A and B? Molecules A and B are positively charged. If they were positively charged, would they be heading to the positive end? No, so we'll get rid of both of those. Molecules A and B are negatively charged. Yes, I agree with that. And then molecule A is smaller than molecule B. Yes, because where is it? Compared to the well, it got to the end quicker, right? So that would be a yes. Okay. Well, I guess that one's not moving. All right. The diagram below illustrates results of electrophoresis of DNA sequences obtained from a family of 2 adults and 3 children and amplified using PCR. The bands represent short repeating sequences of variable length. Results for another female X are included for comparison. Oh, I forgot to mention, when you're running a gel, that you have a ladder, which is going to tell you how long your base pair fragments are. You don't have to guess that. Okay, so here's some DNA from Dad. Okay, let's look at this, put some color on this so we can easily see what we're doing. So there is Dad, there's Dad, there's Dad, and now let's do Mom. Here's Mom, and Mom, and Mom, and not Mom, and not Dad. Okay. Well, yes, there is Mom, and there's Dad. Okay, so let's read. But the banding patterns of the DNA fragments reveal that child 1 and 2 cannot be biological siblings. Are they 1 and 2 no, they are biological siblings, because you can tell they have both mom and dad. And this is very simplified, okay? We're making these statements based on exactly what we see. Child 1 and 3 probably look like the mother. We're not sure of what particular alleles are on these at this time, okay? The mother cannot be the biological parent of all 3. Child 1, she is, but she does not, this child does not have a fragment here, okay? So this is true. The mother's DNA has the same DNA sequence as the father's. Obviously not, because when they were cut with restriction enzymes and then amplified, they have different fragment lengths. And then child 2 and child 3 inherited all their DNA from their father. You can't do that, okay? So the answer is C. Then the mother cannot be the biological parent of all 3. Okay, so DNA sequencing is another genetic engineering technique, and it determines the order of nucleotides in a DNA molecule. It can be used to analyze DNA, identify organisms. They use it in forensics, and you can perform phylogenetic analysis with it. This is activity that I use in my room. It's quite, let me see what my time, I'm going to just run right over this. It has to do with dideoxynucleotides and where they show up as far as in the Sanger sequencing. We have all the A's in cups, T's, C's, G's, and then we pick until we have a DDNTP, and when that ends, we know that our fragment is ending, and then it stops, because a nucleotide cannot be added on to the hydroxyl end, the 3' end of a DDNTP. So you go through this process till you figure out all of the nucleotides of the sequence. We were working with one that was 5 long. And then another thing, this is old school sequencing. Okay, they have NGS, they have Illumina, they have pyro sequencing, all kind of things go on now, depending on the number of the size of the genome, or whether you're doing a fragment, how quickly you need it, that type of thing. So here is old school. We put it in a polyacrylamide gel and then separate these, and these will stop. And then you see that this one stopped right here, and you know that that is a C, okay? And that this one was because it was an A, and then a G, and then a T, and then a C because of location in the tray, okay? And then you can run an electrophoregram, which says the genetic sequence also. But this is fun. If you email me, I will give you where I got this from. I've done it in both of my classes. All right, examples of genetic engineering techniques in analyzing or manipulating DNA. We have a glowing green fish here that has to do with being a modified organism. And then we have some gene cloning. So gene cloning and cloning an organism are two different things. So we have a vector, and then we have, which is a plasmid, that a vector is what we're going to use to get this DNA in. Here's our gene of interest. Both were cut with the same restriction enzyme, and then the restriction enzyme is going to move in to this bacteria and through transformation, and then we're going to, this will reproduce. And if this was reproducing, let's say if this gene was for ampicillin resistant, then the offspring of this bacteria would be ampicillin resistant also. Now, as far as cloning an organism, you take the organism you want to clone, you take a somatic cell, which has all the chromosomes, the 2N, and then you take out of an egg, you're going to take out the genetic material out of that egg, you're going to put those two together, and then you're going to develop a organism that is similar to the first organism. All right, real quick on this, I'll skip that because you can read that at home. Okay, so cystic fibrosis is a genetic condition that is associated with defects in the CFTR protein. The CFTR protein is a gated ion channel that requires ATP binding in order to allow chloride ions to diffuse across the membrane. So the first thing we're going to do is identify the most likely cellular location of mutant CFTR protein that has an amino acid substitution at the ATP binding site. So where would we most likely find this CFTR mutated protein? I'll give you a second. Okay, I hope you're thinking that it's going to be in that plasma membrane, right? Okay, identify again the most likely cellular location of the ribosomes that synthesize the CFTR protein. If we have to get a protein to the membrane, we're going to have to synthesize them on our endoplasmic reticulum, right? Okay, so there's the rope. We'll do that on the ER. And then in the provided model of a cell, draw arrows to describe the pathway for the production of a normal CFTR protein from gene expression to final cellular location. This is where the tricky part is going to come in, okay? So we're going to have to draw some arrows. And so it told us from gene expression. So where does it need to start? It's going to start from the nucleus. It's going to end up at the messenger RNA. It's going to engage with a ribosome at the endoplasmic reticulum. A vesicle will pinch off, take that protein to our Golgi apparatus, and then our Golgi apparatus will take it to the plasma membrane, right? Okay, that's what we say now. All right, so what do we in the plasma membrane? Yes, in the rough ER, and then we went from nucleus to the plasma membrane. Okay, I'm going to address this question one more time in common misunderstandings. All right, so debrief and summary. Difference in gene expression. We've talked about mutations. We've talked about changes in genotype. We've talked about a lot of genetic engineering techniques. It's one of my favorite things to do. All right, so we're back to this question. Now, because there's a mitochondria in this, I graded this question, it was in 2018, so it's one I'm familiar with. So when students saw this mitochondria, for some reason, there were a lot of this happening, and then to there, and then to there. So just because the picture's there, it doesn't mean it's part of it. I think they got confused on the ATP thing, but it said it binds. So a site on that channel actually was where ATP binds, but they got wrapped up in that mitochondria, okay? And then a lot of them said that it would, the lysosome would break it down also. No, we've seen that it gets to the membrane and that it is just non-functional. Okay. All right, I know this is busy. I had a million things to say at the last minute, and I kept adding. So shout outs to kids and teachers that have talked to me over the past 2 weeks, I would say. Some of them I've had a couple of conversations with: Alexa, Sumi, Yule, Sally, Kyle, Patrick, Marie, and Maya today. Well, no, it was yesterday. All right, to-do list. You need to check your device and internet connection. You need to check your email and your phone number, tell your teacher. Keep working on those student practice questions. Okay, keep practicing. There's that symbol. Read the 2020 AP testing guidelines. If you're on your own, your teacher will go over them with you, and we'll also go over them with you next week. But here is the email address to that testing guide, and here is for the practice question. There's the email address, the internet address, and here's the internet address for the exam demo that's working right now. You can start playing with that today, right now, if this is the first time you've heard this. Next week, we're going to have an exam practice week. Kind of excited about this. It's going to be different than what we've usually been doing. So each day of the week, we're going to have something. Monday, there'll be a timed 45-minute window for 2 questions. I'm going to be here with you, but it's going to be like me being your test proctor. I'm going to give you the questions. You're going to write for that time period, and then you're going to save it to the next day. The next day, we'll go over those FRQs. I'll tell you hints and tips about those particular FRQs, that if there are any, and then we'll do I think there's a video on a demo video that we need to watch. And then on Wednesday, you'll do the same thing, 2 new questions, and then you'll go over them on Thursday. And then Friday, Ms. Katay and I will be together. We're going to give you some final test tips, and then we'll have a closing video to show you, and then send you off on your way. And then you're going to just do great. If you've been following us on these videos and doing your practice questions, it's all going to be good. So that's all I have. I know I said a lot of things. I've been going 51, well, probably more than that, probably like 53 minutes. Okay, I'll see you Monday. Bye.